So I'm going to present today some CEH research on nanoparticles, focusing on toxicity, exposure and risk assessment work. This is my outline. I'm going to explain a little bit about what nanoparticles and nanotechnology are, why it's important for society, why environmental releases are potentially important. I'm going to talk a little bit about major past and current CEH funding in this area. Then before I go into some examples of research, I'm just going to talk a little bit about types of nanoparticles, the general CEH research approach which we can identify from our separate projects, and the nanoparticle characteristics that we need to take into account which help us guide our research. Then I'm going to give you some examples, firstly, of exposure and effect research, followed by some risk assessment work, and then I'm going to talk a little bit about what we hope to do in this area in the future. But before I do that, I need to acknowledge not literally the cast of thousands, but the many CEH staff members and students and fellowship holders who've done this work, both in exposure and effects at the top, risk assessment in the middle, and important contributions, chemical analysis, largely Alan here at Lancaster, and Lee Walker helping out with project management. So, what are nanoparticles? Well, the standard definition is a particle of any shape with dimensions in the range 1 to 100 nanometers. For me, as a chemist, it's interesting that it's a physical, not a chemical definition. Nanoparticles can be and are extremely chemically diverse, as I hope to show you. So why are nanoparticles important? Why are they being used so much? Why is their use increasing? Well, it's because basically if you take particles of a substance and make them smaller and smaller and smaller, as you get down into the nanoparticle size range, those particles can take on properties that the bulk material, the larger particles, don't possess, and those properties can be extremely useful. They can be tuned by changing aspects such as the shape of the nanoparticle, and therefore nanoparticles are finding diverse uses in industry and domestic applications nowadays. Just to give you a little bit more idea about relative sizes, this chart goes from 0.1 nanometers on the right up to a very large number on the other side, shows you the size range of nanoparticles highlighted in red against various other small things such as atoms, molecules, viruses and bacteria. And we can see that nanoparticles, they're bigger than atoms, they're generally bigger than molecules except large ones like sugar molecules, and they're in the same size range as viruses, but they are smaller than things such as bacteria and red blood cells. And so while they're particles, they're pretty small, and they're very small compared with the normal size range of particles in the environment, for example. Just to give you another little idea, I put this together. This is an attempt at a scale diagram of a cross-section of a human hair against a 50 nanometer diameter nanoparticle. I hope you can just about see the little red dot highlighted by the arrow there to give you an idea of how small they are compared with something small that you can nonetheless see. So as I said, nanoparticles, nanotechnology have a wide range of current uses. For example, in sunscreens, antimicrobial additive to your socks, medicinal applications, making lightweight boats, for example, and a more recent application in the latest generation of televisions. And the nanotechnology market is expanding. It's been getting steadily bigger for about the last 15 years, and all indications are that it's going to get bigger. There's a great deal of industrial research work on new applications of new types of nanoparticles, and as the years go by, we're likely to see more and more applications and more diverse nanoparticles being used in products. So this is a challenge for risk assessment, this diversity of nanoparticles. Just to come on to some common types of nanoparticles, including the ones that we've used in our research in CEH. Nanotubes, there's a picture there at the top right, are used to provide high tensile strength for materials as diverse as tennis rackets and even boats nowadays. Metals and metal oxide nanoparticles have got a wide range of uses. Silver, for example, is widely used as an antimicrobial agent. The zinc oxide and titanium oxide are used in sunscreens. Cerium oxide can be used in catalysts. Quantum dots, which are semiconductor nanocrystals, are a more recently emerged into the commercial world, and they're now being used in the latest generation of TVs, as I mentioned in the last slide. And as I said, the nanoparticles that we've mostly worked with in CEH are those highlighted here in red, and I'm going to present research involving silver, zinc oxide, and titanium oxide nanoparticles today. As I'll show on the next slide, CEH has managed to get quite a number of significant research projects in nanoparticles, and to me it's possible to generalize what we're trying to do across those projects like this. We're interested in the sources of nanoparticles to the environment, for example here industrial and domestic use, down the drain into the wastewater treatment plant, down the pipe into your river, or into the biosolids which are then spread onto land. So we're interested in where the nanoparticles come from in the environment and how much is getting in. We're interested in the distributions of nanoparticles in the environment once they get there, and we're interested in their effects on biota once they're there. And this is a flavour of our major CEH nano research projects over the last 7 or 8 years. It's not exhaustive. We have also used national capability funding to do some risk assessment, which I've shown you. But it has been a good income stream for CEH over the last few years, and I'd say we're positioned pretty well within Europe and even worldwide as a nanoparticle risk assessment institution. Before I come on to talk about research, I'd like to go into a little bit of detail about nanoparticle types and how nanoparticles may behave in the environment, because these really are key in guiding the research that we do. You can imagine a certain type of nanoparticle, such as silver. It can exist, or it can be made in different sizes, or in different shapes, or it can be made with different chemicals associated with the surface. These are called coatings or capping agents. So a silver nanoparticle can come in many different forms, and therefore it's important to try and see, as far as we can, how these different forms may influence toxicity, for example. I'll show some very nice research about that later. But it just gives you a flavour of the complexity of the nanoparticle field, in that one chemically identical nanoparticle can come in a wide variety of different forms depending on its intended application. The other aspect which guides us in our research is what we know, or what we think we know, about the environmental transformations and uptake of nanoparticles once they actually get out into the environment. This is highly dependent on the type of nanoparticle, but for the ones that we're mostly interested in, metals and metal oxides, processes such as dissolution and loss or replacement of a capping agent, if there is one, could be important in determining the behavior of the nanoparticle and therefore its bioavailability and its toxicity in the environment. And we can construct conceptual models such as this to guide us in the environmental factors which we might want to consider in our research. So now to actually come on to some research, and firstly I'm going to show you some examples of our exposure and effects work. So these are the results of a study which is already published, which we did looking at the effects of a nano zinc oxide on earthworm reproduction in 3 soils which have different pHs but which are otherwise the same. Why do we want to look at pH? Well, we know that normal forms of zinc, what you call the salt or ionic forms of zinc, that their bioavailability and toxicity to organisms in soil is affected by factors such as pH. So immediately we wanted to see whether such factors would also be important in determining the toxicity of nanoparticulate zinc oxide. In this study, as well as looking at the nano zinc oxide, because it could dissolve to form ionic zinc, we also did exposures to ionic zinc itself. And also, because we wanted to see whether there might be a size effect, whether the nano-specific size of our zinc oxide might be important, we also exposed worms to zinc oxide particles that were larger than nano-sized. So what these bars show you for each of those 3 forms of zinc and each of the 3 different pH soils is the concentration of zinc as spiked into the soil, which depresses earthworm reproduction by half. And we can immediately see that there looks to be a pH effect on the toxicity. There's certainly a pH effect on the toxicity of the ionic zinc, that's as we would expect. But we can also see some effects on the toxicity of the nano zinc oxide and the non-nano zinc oxide, which is over there on the right. But also what we can see importantly is that the apparent toxicity of both the nano and the non-nano forms of zinc oxide is lower than the effects of the ionic zinc, that being the concentration of zinc needed to depress reproduction by half is higher in a particular soil pH combination. We can also look at the amount of zinc in the soil pore water, which we get at the point at which reproduction is depressed by 50%. Again, for each form, we still see effects of pH here. But what's most interesting about this is that the zinc in the pore water should represent the dissolved zinc. So in the case of the ionic zinc, it's the zinc that's not adsorbed to the soil surfaces. In the case of the zinc oxides, it could comprise zinc that's created by partial dissolution of the particles. And the interesting thing about this is that if the toxicity that we see here is due entirely to ionic zinc, either what we spiked in or what's dissolved from the particles, we would expect to see the concentrations in the pore water at a given pH to be the same for all 3 forms, or approximately the same. We don't see that. We mostly see that the concentration of zinc in the pore water, where reproduction is dropped by a half, is lower in the zinc oxide exposures, both of them, compared with the ionic zinc exposure. And what that suggests is that the toxicity here isn't just due to zinc that's in ionic form that may have dissolved from the particles. It suggests that intact, undissolved particles remaining in the soils are contributing to the toxicity. So immediately there we have some nice information, somewhat qualitative, but nice information on what the mechanisms of toxic effect of these materials may be. That it's likely to be a combination of both zinc dissolving from the particles, but the remaining particles themselves as well. Both of those are having an action here. We can also look at relationships between dose and toxicity in terms of the amount of metal that's taken up by the worms during exposure, either from soils that are spiked with a silver salt, so ionic silver, or a nano silver exposure. So here we exposed worms to either ionic silver or nano silver, measured their reproduction in terms of the number of young that they produced, and also measured the concentrations of silver in their bodies after the exposure. And this is done for soils here that were spiked with either of these forms of silver, then left for a week before the worms were put in. And the interesting thing that we can see here is that for a given level of reproduction, the worms exposed to the nanosilver have quite a bit more silver in their bodies than the worms that were exposed to the ionic silver. In other words, it appears to be taking a lot more nanosilver to have the same reproductive effect under these conditions. However, if we do the same thing, but instead of leaving the soils for a week between spiking and exposing the worms, we leave them for 2 months, we get this pattern, where essentially the relationship between the concentration of silver in the worm and its reproductive ability looks the same. This is not a function of exposure. In both cases, the worms were exposed to the soils in exactly the same way. It has to be a function of chemical changes that have occurred in those 2 months between spiking the soil and actually exposing the worms. And in this case, the most likely explanation is that that silver has essentially dissolved. So that after 2 months, you're simply exposing the worms to ionic silver that you spiked into the soils, or ionic silver that's dissolved from the silver nanoparticles that you spiked into the soils. So this tells us useful and important information about the potential behavior of nanoparticles if they get into soils. Maybe they don't stay as nanoparticles. What do you do about risk assessment then? We can also use gene expression studies to probe whether there may be subtle differences in the mechanisms of toxicity of types of nanoparticles that have the same chemical core, but have different sizes, or have different capping agents. So in this study, nematode worms, C. elegans, were exposed to one of 4 different types of nanoparticles. Two of them were coated with capping agents that gave the surface no electrical charge, and there were 2 different sizes of those. And there was another one which was, coated with a capping agent that gave the composite a negative surface charge, and a further one that was coated with a capping agent that gave the composite a positive surface charge. If we look at ordination of gene expression profiles, firstly on the left-hand side for the 2 particles of different sizes but with the same capping agent, we can see there's clear clustering of gene expression profiles for chemoreception and transcription. And again, if we look on the right-hand side at the profiles for the 3 types of particles which had the different capping agents but which were all the same size, again, we see clustering. And this suggests that there are subtly different mechanisms of action of each of those different types of the same silver nanoparticle, or the same silver, on the organism in this case. And very interestingly, what was also done was to expose the organisms to a commercial silver nanoparticle from one of our industrial collaborators. And in this case, we know a little bit less about what the particles are like. We know how large they are, and we know they have a capping agent, but we don't really know what that capping agent specifically is. And when you do your ordination, what you see here is that, again, those red points highlighted by the oval, those are gene expression profiles from this commercial particle. And we can see that they are distinctive from all those other particles that we tested. And in fact, they're so distinctive that when you do this ordination, that clustering that you got from the other particles breaks down. So this commercial particle is having quite a different mechanism of effect on the organism compared with those particles that we made. And this is quite important because, of course, essentially we want to risk assess commercial particles. If we don't really know what they are, or if they're having different effects to any particle plus capping agent that we could make in the laboratory, where does that leave our risk assessment? And we've also done similar things to profile the changes in the microbial community structure due to spiking soils with either ionic zinc, a zinc salt, zinc chloride, or zinc oxide nanoparticles. And again, we can see here that the zinc oxide nanoparticles appear to have a distinct effect on the microbial community structure. It differs from that from the zinc chloride. And again, this supports the hypothesis that the nanoparticles themselves do have a direct effect. They're not just dissolving to form ionic zinc and then having an effect. The particles themselves are having an effect. Interestingly here, two different sizes of zinc oxide particle were tested, but they didn't appear to show any difference. Moving on now to a project which I lead called TINE, which is intended to investigate a more realistic exposure route for nanoparticles. And this route is essentially addition of nanoparticles to agricultural soils in biosolids which are produced from wastewater treatment plants. These biosolids will tend to pick up nanoparticles from the influent and concentrate them. They're frequently added to soils as a fertilizer, as a way of inputting nutrients to the soil, but they can also input contaminants such as the nanoparticles. So the idea of TINE was that we would actually produce biosolids that were dosed with either ionic forms of silver and zinc, nanoparticulate forms of silver plus zinc plus titanium dioxide, or an undosed biosolid from a real wastewater treatment plant. And the wastewater treatment plant at Cranfield University was used to do this. So both samples of fresh biosolids and aged biosolids dosed with either nanoparticles or the ionic metal or undosed were produced. And then they were mixed with the soil in different ratios, and we exposed earthworms to them to see whether you would see effects from nanoparticles potentially that have actually been through a realistic process of processing before entry to the environment. These are the results that you see when you expose worms to a mixture of fresh biosolids and soil. So if we look at the orange bars, those represent the lower dose of biosolids. It's a 5% biosolids to 95% soil ratio. And we can see that compared with the undosed sludge, you get a lower worm reproduction rate from the biosolids that have been spiked with either nanoparticles or ionic zinc. And if we look at the red bars over on the right, we can see that we also see that for a higher ratio of biosolids soil. So we're seeing something here. Interestingly, if you look at the green bar over on the left, that's the earthworm performance in an undosed soil. They clearly do quite like the undosed 5% biosolids soil quite a lot compared with the soil itself, probably because it's a good source of food. Coming now to look at the results that we got when we exposed worms to the mixtures of aged biosolids and soils. If you look at the red bars, those are the results from the low dose and a high dose of the nanoparticle spiked biosolids in the soil. And we can see we're definitely getting lower reproduction rates compared with the control, the blue bar over on the right-hand side, the left-hand side rather. The green bars over on the right are a lower dose of either salt or, they are the salt dosed. And we don't see as much of an effect there. And that's very interesting because one might assume that the worst thing that could happen to these nanoparticles is that during the processing would be that they would dissolve up and their chemical form in the resulting biosolids would be the same regardless of whether you started from nanoparticles or metal salts. However, that doesn't seem to be the case here. We don't exactly understand why this is happening. We need to probe the chemical form of the metals in both these types of biosolids more in order to be able to get a better handle on why we may be getting higher effects in the nanoparticle spiked biosolid soils mixtures than in the salt spiked ones. To move on now to look at some risk assessment work. First, I'm going to talk about a little project on aquatic risk assessment of titanium dioxide used in sunscreens, which was funded by National Capability. What was done first was to quantify titanium concentrations in influent, effluent and sludge from a real sewage treatment plant in order to get a handle on what the concentrations and therefore the fluxes of titanium into the environment might be. The interesting point here, about 90% of the titanium is removed into the biosolids, but that remaining 10% does make its way into the final effluent. And the concentrations there can be used as a basis for estimating what concentrations in rivers may be under different conditions. So a number of scenarios were constructed in order to estimate at a regional scale for these two regions of the UK what the surface water titanium concentrations may be as a result of titanium dioxide in sunscreen. So going from scenario 1 down through 2 down through 3, they become gradually more worst case. And scenario 3, the one I'm going to focus on, is basically says you have a one-day heat wave coupled with low flow in your rivers. So everybody goes out and slaps their titanium dioxide sunscreen on, and then everyone has a shower and washes off down into the wastewater treatment plant and makes its way into the river. And because the flow is low, the dilution of the wastewater treatment plant effluent is at a minimum. And when you go through those calculations, you come up with a regional concentration for the Thames region of just under 10 micrograms per litre. So then, that's your exposure. That's your predicted environmental concentration. To assess risk, you need to assess whether you would get an effect at that sort of concentration. And this was done by looking at the combined effects of titanium dioxide particles and ultraviolet light on communities of aquatic bacteria. Simulates the situation in a surface water. You need ultraviolet light in order to activate the chemical reactivity of the titanium dioxide particles, but of course in the surface of a river, and we can assume it's summer, you'll have plenty of sunlight, plenty of UV, so you can assume that the chemical reactivity of these particles will be activated. But what we could see here is that there's actually no observable effect at a titanium dioxide concentration 100 times higher than that concentration of 10 micrograms a litre that we predicted. So we really ought not to be seeing effects, even in a worst case situation like this, at least from titanium dioxide and at least from sunscreen use. So moving on, we also do large-scale spatial risk assessment of nanoparticles in surface waters, and to do this we use the visually explicit GWAVA model. I'll show you some outputs of the model. This is a map of zinc oxide for Europe, and what this shows is the ratio of the predicted environmental concentration of zinc oxide in a particular river reach over the predicted no effect concentration. That's the concentration below which you should not see any effects, and that's derived from ecotoxicological data. If we look at the key and then look at the map, you can see that areas where that ratio is bigger than 1, in other words, where there may be a potential risk, they do exist, they are fairly localized, and they do constitute only a relatively small proportion of European surface waters. I think probably your biggest hotspot is probably around Madrid there. So to come on and think about the future, rather than talk about specific proposals or projects, what I want to talk about is two particular research strands which we hope to incorporate into our work. The first of all is second generation, more complex exposure and risk assessment, where we link environmental compartments such as soils, waters, air and groundwater, and simulate the movement of nanoparticles through them, and also to link that to spatial production and use patterns of nanoparticles to give us more flexible risk assessment tools to be better able to cope with the potential future uses of new types of nanoparticles as they emerge. And the second one is more purely scientific, and it's about how to handle this complexity of nanoparticle types, which I have alluded to a few times. And essentially, we have a hypothesis that you may have all these different types of nanoparticles as manufactured, different capping agents, different sizes, but when they enter the environment, the transformations that they'll undergo, so removal of capping agent, for example, aggregation and agglomeration, will reduce the variability, and therefore the variability in the exposure and effects they may have. And if we can show that there's a reasonable hypothesis, that'd be really important for making risk assessment of all these different types of nanoparticles, including ones that haven't been made yet, far more manageable for policymakers. So with that, I'll say thank you very much.